In this video, I'm going to review carbohydrate biochemistry, and I will attempt to highlight those particular aspects which are frequently seen on the step. And so we'll begin with the glycolysis pathway, and then we'll deal with part of the pentose phosphate pathway, and we'll deal with the regulation of PFK1, and then we'll take a look at the TCA cycle and the electron transport chain, and the shuttles, and maybe even glycogen storage disorders, and the aldose reductase pathway, which is very important for the step. Okay, so glycolysis begins with glucose, which is converted in the cell to glucose 6-phosphate, which is essentially trapped in the cell, a process known as ion trapping. The glucose 6-phosphate is then converted to fructose 6-phosphate, which in turn is converted to fructose 1,6-bisphosphate by PFK1, phosphofructokinase, which, as I said, is a highly regulated enzyme. And the fructose 1,6-bisphosphate is split by aldolase into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The glyceraldehyde 3-phosphate continues in the pathway to form 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, and then the high-energy compound phosphoenol pyruvate, which in the end is converted to pyruvate. Now, once the pyruvate is formed, it can be converted to acetyl coenzyme A. If NAD plus is available and the various cofactors available, or under anaerobic conditions, it's converted to lactate through lactate dehydrogenase. The conversion of pyruvate to acetyl CoA is carried out by the pyruvate dehydrogenase complex, or PDH. Note that NAD plus is required for this step of glycolysis. And now I wanted to very briefly show you the regulation of PFK1, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. And essentially, this PFK1 enzyme is regulated by the product of another enzyme known as PFK2, phosphofructokinase 2, which synthesizes fructose 2,6-bisphosphate. And fructose 2,6-bisphosphate is a positive allosteric regulator of PFK1. And so formation of fructose 2,6-bisphosphate will activate PFK1. And if we take a look at the regulation of this enzyme, insulin, for example, will activate PFK2, resulting in increased formation of fructose 2,6-bisphosphate, which in turn will activate PFK1. Glucagon, on the other hand, has an inhibitory effect on phosphofructokinase 2, PFK2, and therefore less fructose 2,6-bisphosphate is formed, therefore less activation of phosphofructokinase 1, PFK1. That's, of course, because glucagon wants to keep the glucose levels elevated, whereas insulin wants to promote the metabolism of glucose. Now, in this slide, I briefly want to show you the interaction between the glycolysis pathway and the pentose phosphate pathway. And the intermediate formed in glycolysis, glucose 6-phosphate, this can be converted into pentose phosphate pathway to 6-phosphogluconolactone, which through various steps will eventually end up as ribose 5-phosphate, which is used in purine and pyrimidine synthesis. Now, this conversion of glucose 6-phosphate to 6-phosphogluconolactone is carried out by G6PD, or glucose 6-phosphate dehydrogenase. And in this very first step of the pentose phosphate pathway, we generate NADPH, And you'll notice NADPH is also generated in step 3 of the pentose phosphate pathway. This NADPH is used in fatty acid synthesis, and also very importantly, the NADPH is a very important enzyme for the red blood cells. So in the absence of glucose 6-phosphate dehydrogenase, there's not enough NADPH formed, and the NADPH cannot keep glutathione in the reduced state. And if that glutathione is not reduced, it makes the red blood cells fragile. This results in hemolysis and jaundice. And drugs which can precipitate deficiency of this enzyme are sulfonamides and some antimalarial drugs. So do remember this, it's pretty high yield. Now, the pyruvate that is formed from the glycolysis pathway, this is converted to acetyl coenzyme A, as I mentioned earlier, using the enzyme PDH, or pyruvate dehydrogenase. It's an enzyme complex requiring a number of cofactors, one of which is vitamin B1 of thiamine, which is often deficient in alcoholic patients. And so once pyruvate is then converted to the acetyl coenzyme A through the PDH, acetyl coenzyme A carries 2 carbons, of course. That condenses or reacts with oxaloacetate, which is a 4-carbon compound. And this reaction results in the formation of a 6-carbon compound, which is citrate. And so the formation of citrate in this reaction is catalyzed by citrate synthetase, and it's a very very regulated enzyme, and you should look at certain aspects of its regulation. But citrate, being a 6-carbon compound, is converted to isocitrate, and isocitrate, through a decarboxylation reaction, is converted to alpha-ketoglutarate. The alpha-ketoglutarate, in turn, through another decarboxylation reaction, is converted to succinyl-CoA. So you'll see that on either side of alpha-ketoglutarate, we have a decarboxylation reaction, and this results in succinyl coenzyme A, which is a 4-carbon unit. This is converted to succinate, and then to fumarate, and then malate, and then back to oxaloacetate. Now, the 2 carbons that were originally added from acetyl-CoA are still present in the oxaloacetate molecule. They will only leave as carbon dioxide in the next cycle, when 2 more carbons are added onto oxaloacetate from acetyl-CoA. So it's like an airport with planes landing on the one side and planes taking off on the other side. So these carbons added from the acetyl group only leave in the next cycle, and so the entire molecule is eventually oxidized. So, so take a look at this carefully, and you'll see that 3 NADHs are produced in the cycle, including 1 FADH2, which will enter the electron transport chain. So that takes us to the mitochondrion now, and the mitochondrion, as you know, has an inner membrane and an outer membrane. The inner membrane has the components of the electron transport chain. And in this process, various electrons are transferred from the NADH and FADH2 formed in the TCA cycle. And this leads to the generation of energy, which is used to pump protons into the intermembrane space of the mitochondria. These protons eventually can enter the mitochondrial matrix uh, yeah, through the complex 5 with the generation of ATP. So this is complex 1, which uses NADH, as I showed you, generated from various reactions in the TCA cycle. And complex 2 is actually succinate dehydrogenase from the TCA cycle. And you'll notice complex 2 is not a transmembrane protein. And so complexes 1, 3, and 4 are responsible for the transfer of protons from the mitochondrial matrix into the intermembrane space. And so that the intermembrane space becomes acidic, it's highly charged with protons. And these protons can only move back into the intermembrane space through complex 5, which is then coupled to phosphorylation of ADP to form ATP. So in complex 4, you'll notice that oxygen acts as the final electron acceptor to form water. So the electron transport chain is essential for the formation of ATP for the cell, and we would not be able to survive without that. But there are a number of agents which can inhibit the electron transport chain. For example, complex 1 is rotenone. Complex 2 is malonate, which inhibits succinate dehydrogenase, which, as I told you, is also the same enzyme in the TCA cycle. Okay? Complex 3 is antimycin, and complex 4 inhibitor is cyanide. And then we have complex 5, and the inhibitor there is oligomycin. And one must remember that the two main units of complex 5, also known as ATP synthase, these are held together by a protein which is known as the oligomycin sensitivity conferring protein. And so it is this protein that is inhibited by oligomycin. So do remember those. And then we also have the uncoupling agents or uncouplers, which also disrupt the electron transport process. These uncouple electron transport from oxidative phosphorylation. And so one very good example is the compound known as 2,4-dinitrophenol, which is used in the explosives industry. And what these uncoupling agents do is that they bind the hydrogen ions in the intermembrane space, and they move these into the mitochondrial matrix. So this is not accompanied by the generation of any ATP. However, the transfer of the hydrogens back into the intermembrane space results in the generation of heat energy. So body temperature increases, oxygen consumption increases, but there is no accompanying synthesis of ATP in this process. So 2,4-DNP is also illegally used as a weight loss agent because it causes thermogenesis, but many people have died from its use. So in addition to uncoupling agents such as 2,4-DNP, we also have proteins which are located on the inner membrane called uncoupling proteins or UCPs. These proteins allow the passage of hydrogen ions into the mitochondrial matrix, again with the liberation of heat energy, but no accompanying synthesis of ATP. Yeah. So those are the uncoupling proteins, and there are a number of different uncoupling proteins that have been identified. So now you know the inhibitors of the electron transport chain. as well as the agents such as 2,4-DNP, which is an uncoupling agent, and the role of the uncoupling proteins. So please make sure you know those. And this particular slide, I want to show you what happens to the NADH, which is produced in the glycolysis pathway. Now, the NADH is not permeable to the mitochondrial membranes, but we need to transfer that energy from NADH produced in glycolysis. And the question is, how do we get that NADH into the mitochondrial matrix? So we use one of two shuttles. One is called the malate aspartate shuttle, which is being depicted in this particular slide. So remember, we want to get the NADH from the glycolysis into the mitochondrial matrix, where it can participate in complex 1 of the electron transport chain. So the cell does a very clever thing in that it uses this malate aspartate shuttle, and in the very first step, you'll see that the amino acid aspartate, this is converted to oxaloacetate, as you will see shortly. And the oxaloacetate is then converted to malate, as you'll see on the top right-hand corner of this slide. And the conversion of oxaloacetate to malate will require the NADH produced in the glycolysis pathway. So those hydrogens from NADH are now trapped in malate. Malate has the ability to cross the inner mitochondrial membrane through a transporter. And so you can see the malate coming through. It is now present in the mitochondrial matrix. That malate is now converted back to oxaloacetate. And in so doing, it transfers its hydrogens to oxaloacetate. Transfers its hydrogens back to NAD+, which is inside the mitochondrial matrix. And now we have reformed our NADH. The oxaloacetate is converted back to aspartate, and the aspartate can now leave through its transporter. And so essentially what we've done is we've transferred the NADH from glycolysis into the mitochondrial matrix by using this shuttle. Inside the shuttle, we have the glutamate donating its nitrogen to form alpha ketoglutarate. And you can see that process occurring in the mitochondrial matrix as well, where the glutamate transfers its nitrogen to oxaloacetate to form aspartate, resulting in alpha ketoglutarate. So there you see in the bottom right the NAD plus being converted to NADH. And on the top there, you will see the NADH donating its hydrogens to oxaloacetate. And the second shuttle is the glycerol phosphate shuttle. And this particular shuttle operates mainly in brown adipose tissue. And this is because brown adipose tissue is mainly present in newborns. And The energy from this pathway is used for thermogenesis, and it keeps the baby warm. So here in this shuttle, you'll see NADH. The NADH then donating its hydrogens to form dihydroxyacetone phosphate, and these are picked up by FAD to form FADH2. The FADH2 goes to complex 2 of the electron transport chain and will only generate 2 ATPs instead of the 3 ATPs generated from NADH. And because of that, the additional energy is used as body heat. We now deal with the glycogen storage disorders. Just to rapidly take you through these, the high yield ones are von Gierk disease, which is a deficiency of glucose 6-phosphatase. It also results in gout in some patients, hyperuricemia. Type 2 is Pompe, alpha-1 for glucosidase. Type 3 is Cori disease, absence of a debranching enzyme. And the amylo-1,6 glucosidase. Type 4 is Anderson's disease. And type 5 is McArdle's, which is very high yield. You'll always find the vignette saying that the patient complains of muscle pain after exercising. And then the urine is usually red in color due to myoglobin. And McArdle's is due to deficiency of glycogen phosphorylase. So type 1, type 5 are pretty high yield. And then there's type 6, 7, and 8. In this, this slide, I want to point out a few important steps in gluconeogenesis, which you are likely to see. And if you can read through these, remember these pyruvate carboxylase is the enzyme that converts pyruvate to oxaloacetate. And that, of course, reaction requires the presence of biotin. And then there are a number of other steps involved in gluconeogenesis, if you can read through those. And the important thing is to remember that biotin is essential for these decarboxylation reactions. This aldose reductase pathway, or the polyol pathway, is also very important for the step. Remember that when glucose concentrations are very high, this activates the aldose reductase enzyme, which then converts glucose to sorbitol, which in turn is converted to fructose. And this enzyme, as I said, is activated in the presence of high levels of glucose. It has a high Km and low affinity for glucose, and it's implicated in a number of complications associated with diabetes, such as microvascular